Hello, and thanks for joining us today for the latest Technology Networks webinar, Fast and Simplified Biopharmaceutical Characterization. I'm Jack, Senior Editor for Technology Networks, and I'm here to moderate the event. I'm pleased to have Sean McCarthy joining us today as your presenter. Sean received his degree in chemistry from the University of Vermont in 2005, followed by an NIH postdoctoral associate within the Department of Pathology at the same institution. His work centered on environmental oxidative stress-related diseases using a variety of biochemical and mass spectrometry-based techniques. Sean has worked in the mass spec industry since 2008 and held several scientific and business development positions to address pharmaceutical and biopharmaceutical characterization. Sean is currently the Global Market Manager of Biologics at SCIEX, where his focus is on delivering targeted solutions for biopharmaceutical characterization. Warm welcome to you, Sean. Following the webinar, we will have a Q&A session, and we would welcome any questions that you may have. To ask a question, you should enter your question into the box on the left-hand side and click Submit. We will answer as many as possible during the time available. Any questions we don't have time to get to, we'll be sure you're contacted offline with an answer. If you experience any technical issues, please use the question box to request support. Please remember you can ask questions at any time, even whilst watching the webinar on demand. I will now hand over to Sean.
Thank you very much, Jack, for the introduction. As Jack mentioned, I want to discuss some recent work we've been doing to address the area of routine biologics characterization. So in terms of an overview, I'd like to give a brief overview of some recent advancements we've made in both our hardware as well as the software required for collecting as well as processing the data. Then I want to move into discussion of some different workflows that we offer with this solution and discuss them in some detail, including doing intact mass analysis, antibody-drug conjugate, drug-to-antibody ratio calculations, subunit analysis, peptide map, and then at the end of the presentation, give a brief summary. So at the start, I'm happy to introduce something we just recently launched, which is the X500B from SCIEX. So it's the X500B QTOF System. And there really are three different components to this instrument. One is the front-end separation, which is afforded by the Exion LC system. The X500B QTOF system, which I'll be discussing in more detail as we go forward. And then the software, both SCIEX OS, which I'll describe in a few moments, is really a new software platform which is extremely intuitive and very easy to use, and also BioPharmaView for doing the processing of that data ultimately. So in terms of speaking about the X500B, one of the questions we've had at the outset is, what's the big deal with the instrument? So you can see here, this is sort of looking at a value map versus the amount of effort it takes to bring a new platform to the market, as well as tracking the lifetime of that product. And you can see that essentially every technology has sort of this characteristic S-shaped life cycle, where there's some initial challenge in getting that platform to the market. There can be some improvements, but at some point technology starts to advance to a point where improvements on that same platform become a significant challenge. So, bringing new technology to the market does require a significant amount of effort, and the amount of effort required really is driven by the amount of disruption that you're bringing, and that's what I want to discuss with what we've introduced on the new platform. So the X500 does represent our next generation mass spectrometer. So it's a platform architecture that was the most complex that we at SCIEX had taken on since the API 300 back in 1995. And at any time during the development of this product, we had as many as 70% of the hardware R&D resources and 60% of the software R&D resources working on SCIEX OS, which is the informatics system for controlling the instrument. As part of the development, we used a lot of predictive modeling and a lot of testing to enhance the reliability and test in the reliability of the platform. And we also invested a significant amount of time in the design and usability of the instrument, not only of the X500B itself, but also in the usability of the software to control the instrument to ensure it would be approachable for a wide range of users. To discuss to some extent some of the novel technology that is in the X500 platform and which lends it to being a very complex, a very compact instrument for the market. We've introduced some novel optics. So you can see here on the left-hand side of the slide a novel end geometry. So this is distinctly different from other TOF platforms on the market today. And with the introduction of this end geometry, we're able to really reduce the overall size of the TOF flight tube. And in addition, the design also incorporates some advances which reduce the number of voltage settings and other types of settings that are common on a TOF platform. So it does make it much easier to set up and run and optimize, but also reduces the overall size of the instrument as a whole. In addition, in looking at some of the front-end ion optics, we've also introduced and incorporated into this platform triple-stage turbo pump. So it's a single turbo pump that can accomplish the achieving vacuum all the way through the front-end ion optics. So again, reducing the complexity of the platform and making it much easier to service at some point if that's required. Some of the other things about the instrument, so as I mentioned, it really did represent a very complex and comprehensive development of a QTOF platform. So roughly 5% of the instrument has been reused from previous generation platforms. And a large extent of that is really on the front end, still incorporating our Turbo V source. So that's where a lot of the reuse has come from. But as I mentioned before, it's really been engineered for simplicity with the NTOF geometry and reducing the number of voltage settings required for optimal performance. We've also introduced a twin spray emitter, which has separate channels for both your calibrant as well as your analyte, and that's designed to minimize any crosstalk between your calibrant and your samples that you're analyzing. Something that shouldn't be understated is the footprint, overall footprint of the instrument. So it is quite compact, and overall, not only the footprint of the instrument, but the overall height of the instrument with the novel NTOF geometry, it really does represent a very compact yet high capability TOF platform. It's also been designed for very easy service, so making it very simple to clean at some point if that becomes required. And also it has integrated calibration solutions on the instrument. So there's no need for external syringe pumps or filling of syringes. Now you can have reservoirs of your both positive and negative ion calibrants on the system as required. As I mentioned, the other innovation that we've made with the introduction of this platform is the development of SCIEX OS. So SCIEX OS represents the next step in our software that takes the stuff from Analyse and incorporates the capabilities of both MasterView and Multiquant and incorporates them into an overall acquisition package, which is was first introduced on our ARP platform just a little over a year and a half, 2 years ago. And it does represent a significant step in ease of use. And I'll show that. Show that here. So you'd see familiar with the previous software. This represents really a more modern type of interface, which is more in line with the ease of use that our customers have been asking us for. You see, it's very tile-based, where you can define both the parameters for your acquisition for what samples you're going to analyze. So what does your sample list look like, as well as the LC and the MS method and all of the tuning parameters. And then also incorporation of all of the necessary analytics to move forward after you've done your acquisition to process and report your data. And we also have incorporation, and I'll be discussing in a few more moments, a tile to access BioPharmaView for doing the biologics characterization as well. The interface itself can also be customized to allow either access or remove access depending on a user's rights or access capabilities on the platform, so it is quite flexible to allow some customization there as well. And you can see on the right-hand side, we've also incorporated sort of a dashboard for the readiness of the overall system for doing the analysis. So it's a really quick and easy to use interface. It's very tile-based and extremely intuitive to use. All right, I need to stop. So in addition to the SCIEX OS and the improvements we've made there in terms of the ease of use overall of acquiring the data, we've also been working over the last several years to introduce software specifically for processing biologics data. So that's in BioPharmaView. So BioPharmaView, the most recent release is our 2.0 software. So overall, building on our previous versions, it is very simple to use and powerful for doing that more routine biologics characterization. And it does do that processing and the analysis extremely quickly, making it easy to get from your data to the correct answer in a short amount of time. It is designed to handle batches to do more assessment of comparability between different samples. And the software is also quite flexible, where there are a large number of built-in modifications, but if the particular modification you're looking for is not already in the software, it's flexible enough to allow you to define those additional modifications. It does also support both IDA as well as swath acquisition, which we discuss in a few more moments, but does support both of those different workflows. And supports the main key routine workflows. So that would be, intact protein, which would include the subunit analysis, as well as peptide mapping, both doing reduced and non-reduced peptide mapping, so the ability to do confirmation of disulfide linkages. Antibody-drug conjugate, drug-to-antibody ratio calculations have also been incorporated, and as I mentioned before, the ability to do comparability between different samples is included in the software as well. And I'll show that as we move on. So as I mentioned, BioPharmaView is extremely easy to set up. So I wanted to show, at least at a high level, some of the key areas in which you can enter information to start progressing through your analysis and to walk through each of those components separately. So the first is, you can see there's a very logical tree on the left-hand side, which basically describes different steps within an intact and a peptide mapping workflow, where you can characterize your standard as well as do comparisons between different samples. So a very easy, logical way to walk through those experiments. The next is an area where you can define the sequences for both, in the case of an antibody, like we show here, the heavy chain and the light chain, or alternatively a different biomolecule where you may have either multiple chains that may be different or maybe a single chain that would be the same. The flexibility to define the sequence of your biologic within the software quite easily, and being able to find multiple chains of that as well. Next is an area where you can define your modifications. So as I mentioned in BioPharmaView, there are hundreds of different modifications that are already built in, but this is also a place where you can custom define a modification depending on the type of analysis that you're doing or the type of molecule that you have. Next is where you can define your disulfide linkages that would be either intra or interchain, and those can easily be defined here. And it's very nice with the software. We'll keep track of the cysteine residues that you've already used to define those disulfides. So once they've been used once, there's no risk of using them again. And then lastly, we have the ability then to define the specific parameters that you would use for both a peptide mapping as well as the intact mass workflow. So you can define different parameters for how you're going to treat each of those sets of data, but again, using the same underlying sequence, modification, and disulfide data. So there's no need to have separate experimental files for each workflow. You can use the same information and define, depending on the type of experiment, which workflow you're going to use and what considerations you're going to take for each of those. So now I would like to move on and discuss actually the use of both the Exion LC for the separation, the X500B for the data collection, and then SCIEX OS and BioPharmaView for the analysis, and sort of discuss and show some examples of the different workflows in the work that we've done. So I'll start first with some intact mass and some subunit analysis. So as I show here, is an example where you would, in BioPharmaView, characterize a single biologic where you have on the top right-hand pane. In BioPharmaView, you have the total ion chromatogram for that analysis. Underneath that is an extracted spectrum, which the area over which you extract that spectrum is defined as part of your processing method. And then underneath that, on the bottom right, is a reconstructed spectrum. And each of these windows can be expanded, and you can zoom into the spectrum as required. So you can see very easily on the right-hand side, you have access to all of the different stages of your data. On the left-hand side, you have some of the parameters, a subset of the parameters that you defined for your experiment. So there's some flexibility to fine-tune those parameters. And then underneath that are all of the identified modifications based on your input for the experiment. And you get both the expected mass as well as the determined mass, the relative percentage, and a description of what the modification that has been identified is for each of these. And you can toggle and interact with those modifications as required. As I mentioned before, one of the powers of BioPharmaView is the ability to then batch analyze many analytes. So you can take the parameters that you've used to characterize your main, say your standard in a particular experiment, and then use those same parameters and compare different samples. So one of the views in doing that comparison is shown here, where on the right-hand side we have the representative spectra, both the raw and the reconstructed spectra. In this case, we've hidden the chromatogram. And we also have a graphical view looking at a bar chart. And this is looking at the relative abundances of each of the different modifications across, in this case, 2 different samples. So a very easy way to do a comparison between 2 samples and identify which species are present and how different those species are from each other. So often one of the questions, also looking at doing these analyses to say, look at a batch of experiments is, how robust is the platform? And it can be a challenge to prove how robust something is. So this is one of our, one example we've done to sort of look at how reproducible and robust the instrument is. So this represents an analysis that we've done over a period of 25 hours. So that's what we've shown here in the top right is a plot of the mass error for some different glycoforms within an antibody over the course of that 25-hour experiment. And what's key to note here is that overall the mass error is roughly plus or minus 10 ppm, but this is based off of a single calibration at the start of the experiment. So there was no additional recalibration. And the reason for the mass stability really goes back to the design of the X500 platform, where it has a heated TOF area, which really maintains that calibration and gives a reproducible result over an extended period of time. The spectra that are shown here are an overlay, in this case, of one analysis for every hour over the entire 25-hour experiment. You see it's extremely reproducible at both the full spectrum as well as a zoomed in of the different charge states across that entire experiment, and highlighting the reproducibility and robustness of doing this type of analysis on the platform. I mentioned drug-to-antibody ratio calculation is something that's incorporated in BioPharmaView. So again, very similar to the intact mass, where you have your total ion chromatogram on the top right, your spectrum as well as the reconstructed spectrum are shown. Something that is also displayed here is on your reconstructed spectrum. If you want to find the evidence for a specific component that's identified, you can select that component, and then you can see in a trace above that, sort of the middle right, all of the vertical red lines. Those are the indications of where the reconstruction has seen evidence in the raw data and then converted it into the reconstructed. On the lower left-hand side, you can see a bar chart, which is a plot of each of the different drug-to-antibody ratios, so whether there be 2 or 4 or 6 drugs, depending on what specific molecule you're looking at, and then an overall calculation of the drug-to-antibody ratio, which again is automatically calculated within BioPharmaView. Finally, looking at subunit analysis, so that's something that's obviously supported within BioPharmaView. So this is looking at just a heavy chain of a monoclonal antibody, but very similar to what we have seen for the intact. In this case, highlighting the ability to do that comparability, so doing a mirror plot of 2 different spectra at both the raw as well as the reconstructed level, and then also further highlighting something I demonstrated earlier, which is the ability to look at a bar chart, to look at each of the features of the molecule over a set of analyses, and really look and assess at where the differences are between 2 different samples. So from there, I'd like some work we've done with peptide mapping as well on the X500B platform. So as I mentioned earlier, we offer 2 different acquisition modes for doing peptide mapping. The first is really doing an IDA type of acquisition, where you'll do a precursor scan and then take maybe the top 10 or 15 of those most intense precursors and then select them for subsequent MS-MS analysis. Another way in which we often do our analyses, really to give a much more comprehensive view and also allow us to start doing more quantitative analysis, particularly at the MS-MS level, is using SWOT acquisition. So SWOT acquisition is a way of acquiring data where you use portions of a quadrupole mass window. So you have a window of masses that changes from throughout the course of a, say, for lack of a better term, throughout one Q1 scan, and you pass narrow windows of mass through to be fragmented, and that gives you much greater correlation between a precursor mass as to its MS-MS spectra, but in addition allows you to really dig deep into every component that's detectable within your sample. So it's a much more comprehensive way that allows you to really narrow down which fragment ions correlate to which of the parent ions. And both of those workflows, as I mentioned, are both supported on the X500B platform as well as in BioPharmaView. So if we look at applying a peptide map type of workflow and using that in on the X500 platform as well as in BioPharmaView, you'd see we can do our peptide map analysis, and this actually is quite fast. It's hard to demonstrate in a webinar how quick the analysis is, but it is a very quick turnaround. But again, similar to the intact mass, we have our same file where we've defined the sequence as well as the modifications and all the disulfide linkages. And we can easily process that data. And we can see our total ion chromatogram as well as the relative abundance, sort of in the third pane down, for each of the modifications that we've searched for in our data set, as well as on the bottom left, our MS, and on the right-hand bottom right, the MS data for a particular selected peptide. We also get a very good view of the overall sequence coverage for our analyte. We can see high sequence coverage throughout the experiment. It's similar to what we had seen on the intact mass level. One question comes up is in terms of mass accuracy, particularly across a sample and then also across samples. So showing here that we have very good mass accuracy for a variety of different peptides within a single analysis, and we've also seen that across different analyses, again highlighting this mass stability of the X500B, and also showing here, similar to the intact mass workflow again, where you have the ability to display and do a comparison between samples by displaying mirror plots for the chromatogram as well as the raw and the MS and MS level data for a peptide map. In terms of looking for critical quality attributes or modifications within your peptide map, that is quite simple. So you have the list of all of the different peptides, and each of the different forms of that peptide are different entries. So if you had for example, a non-deamidated and a deamidated peptide, they would be separate entries, and you can certainly view them and compare them to each other, both within the same sample as well as across samples, to start doing an assessment for how much those critical quality attributes are changing, and also looking for specific critical quality attributes that are important for the analysis that you're doing. So showing you a couple of examples, in this case looking at the oxidation of a methionine residue. So we can easily select both the unoxidized as well as the oxidized form of that peptide. And then if we dig down into the data, we can very easily do a comparison all the way down to the MS level, where we see here high quality MS data for each of those. And we can very easily confirm where within the peptide the oxidation has occurred by looking at where the mass shift between those two peptides has occurred, to really localize where the modification is. And again, this is something that is automated in terms of identifying that peptide is modified in BioPharmaView, but also allowing easy access to the underlying data to do more of a manual confirmation if that's something that's required. Similarly, looking at yet a different type of modification, something that could potentially be even more complicated, in this case a deamidation, where we have a 1 Dalton mass shift. Again, we can look at each of those peptides, overlay the MS spectra, which are clearly shifted by 1 Dalton in mass, and then confirm that at the MS level. Even though one of these peptides is at low abundance, still getting very high quality MS data, and that's really coming from our ability to use a swath acquisition to really narrow into those narrow mass windows and get high quality MS data even on low level types of modifications. And finally, one of the areas of doing a biologics analysis, or really any analysis, whether it be in biopharma or otherwise, is the ability to report the data. The ability to collect it, process it is one thing, but you also need to communicate that within your organization. So we've offered in BioPharmaView a very good and robust way to do that reporting, where we can report out not only the experimental information that defines how we've collected our data and what the parameters of that are, but we can also report out the specific information about the different critical quality attributes that we were looking for, as well as the different spectra that correlate to that raw data. So in this case, looking at an intact mass at both the raw and the reconstructed level. So very flexible reporting to allow you to report out what you need within your organization. So with that, I'm drawing to a close in the presentation, but I wanted to take a few moments just to highlight that what I've discussed today is really the X500B as well as SCIEX OS and BioPharmaView, and how taking those three components together really does lead to a very streamlined solution for doing biologics characterization. However, those are not the only solutions that we offer for biologics characterization. We also have a separation for doing CE, so either standalone CE or a CESI type of interface for doing more capillary electrophoresis types of separations, which is a very powerful technique for both doing glycan analysis and maybe assessing charge heterogeneity within a molecule in a very unique way. So we offer that capability as well as our service and support and our commitment to ensuring that our customers are successful in implementing their solution. And we offer, again, even more advanced types of mass spectrometry with our other triple TOF platforms, which can really expand and allow you to do some of those advanced characterization techniques, which you may need to do. So with that, I'd like to close and so with that, I thank you for taking the time to listen to this today, and I'll hand it back to Jack. Thank you.
Thank you very much, Sean. I'm sure you will all agree that there were some excellent points raised during the presentation. We've had a number of questions, so I'll move straight on to these. In the presentation, you mentioned that you can define disulfides. Does your solution allow you to search your data for these? And what about scrambled disulfides?
Yes, so certainly we have the ability to search for the expected disulfide. So whichever disulfide linkages have been defined as part of the analysis, we can search for those as part of our normal peptide map processing, a non-reduced peptide map. In terms of doing the scrambled disulfides, that's not currently a capability that's within our software, but certainly something we understand is important in the industry and something we're working towards implementing in future versions of the software.
Thanks, Sean. That's great. Just to remind everybody that it's really easy to ask a question, so if there's anything that you'd like to hear about from Sean, just type your question into the box and press submit, and we'll get straight on to that. Sean, the next question that we've had is the software 21 CFR compliant, and does it meet FDA data integrity regulations?
So currently the software is actually not 21 CFR compliant ready. So we understand that that's, particularly in the biopharm industry, there's a particular movement towards implementing MS further downstream and in different processes. So we understand that there's a requirement there, and that's something that we're actively working towards. Given our experience working in regulated environments, we do have some experience implementing that, and that's something we're working towards having both in SCIEX OS for data collection as well as in the associated software packages that would be required for doing ongoing analysis.
Thanks, Sean. Moving straight on to the next question. Somebody's asked, what is the resolution of the X500B?
So the resolutions, it's often a challenge to quote specific resolution because it depends on what the particular analyte is. But the specification on the instrument for looking at one of the analytes that's biologically relevant that we use for specification on the system is bovine insulin. The +6 charge state of bovine insulin has a resolution of 42,000, and that's the specification. Often we can go a little bit above that, but that's what we have in our specification sheet.
Thanks, Sean. That's great. Moving straight on again to our next question. You showed examples looking at components over a number of samples. Are you able to select the components you view?
Yes. So it's very the software is very flexible, where you can, whether it be for an intact analysis or for a peptide, you have the ability to sort the data. So if you're looking for a specific component, so you're looking for a particular glycan structure, or you want to see all the peptides that are, say, modified by deamidation or some other modification, you can sort the data that way. And then also, when you look at the bar charts that I had shown in several of the slides, you can also define which of the components in the samples would be plotted in those different bar charts. So you can both sort the tabular data as well as the graphical data to look specifically at which attribute or modification you're interested in, and report out the plots in that same way in the final reporting as well.
Thanks, Sean. Just so everybody knows, we've got Sean available for a little bit longer, so if you've got any questions, please do send them in. So moving on to the next question that we've had. I'm not analysing antibodies, so can I use BioPharmaView software for the analysis of other classes of biopharmaceuticals?
Yes, certainly. So yes, all the data that I had shown was on on monoclonal antibodies. So, which, as I understand, are fairly, from a biologic standpoint, are actually quite simple molecules. BioPharmaView offers you the ability to define whatever protein sequence, as well as any other modifications. So more complex molecules would be, you'd certainly be able to analyze, and even those which may be, say, bispecific or a dual variable domain or something, where you may have different sequences within an antibody, you can define each of those sequences separately as well. So it does give great flexibility for a wide range of sort of amino acid-based types of biologics. What we don't currently support in BioPharmaView are biologics, such as, oligonucleotides or things like that, things that are not peptide or amino acid-based analysis. So as long as it's a peptide-based therapeutic, BioPharmaView is certainly flexible enough to handle and process that data.
Thanks, Sean. The next question that we've had is from Larry Mallis. He's asked, when will SCIEX OS be available for triple quads? What will the upgrade path be for currently owned systems?
So that's a question I don't actually have a concrete answer to. So what I can say is the SCIEX OS represents the next step in our software, and certainly a goal is having SCIEX OS for our tandem quadrupole instruments as well. The time frame over which that will be implemented, I don't think I could give a rock-solid number on that, nor could I really define what the upgrade path would be. But certainly, I think the point is well taken that people that we've shown SCIEX OS to, it is a dramatic change from what people may be used to in, say, Analyse in terms of ease of use. So we're certainly aware that there's a desire, and we're moving forward towards having SCIEX OS control more and more of our instruments in the future.
Thank you very much, Sean. Unfortunately, that's all we have time for today, but just to remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. Also, you can continue to ask questions even when watching the webinar on demand, so please do that. The webinar will be made available on demand very shortly, so if you have any friends or colleagues that you think may be interested in this webinar or any of our future events, please feel free to share the link. Thanks again for your time, everybody.